Hi friends, welcome to Science Jagat. So today we are going to talk about Recombinant DNA Guidelines 1990 and its recent updates. So Recombinant DNA Guidelines in 1990 advances research in biotechnology, which is initiated by Indian Institutes and Industries. DBT, which is a nodal department for promoting biotechnology program and provides scientific support for biosafety, had formulated recombinant DNA guidelines in 1990. So these guidelines are further revised in 1994 to cover R&D activities on GMOs, transgenic crops, large-scale production and deliberate release of GMOs, plants, animals and products into the environment, shipment and importation of GMOs for laboratory research. For research, the guidelines have been classified into three categories based on the level of the associated risk and the requirement for the approval of competent authority. So let's see what are the categories. So category 1, activities include those experiments involving self-cloning using strains and also interspecies cloning belonging to the organism in the same exchanger group or in the same species, which are exempted for the purpose of intimation and approval of competent authority. So this category involves experiments which have low risk. Fine. So this is from the biosafety level 1 and these categories does not require any kind of approval. Then comes category 2. Activities which require prior intimation of competent authorities and include experiments falling under contamination level 2nd, 3rd and 4th. So details of each contamination level have been provided separately in the guidelines and in very details. So this kind of category needs prior approval before starting your experiment and before taking your product to the market. Then comes category 3. Activities that require review and approval of competent authority before commencement include experiments involving toxin, gene cloning, cloning of genes for vaccine production and other experiments as mentioned in the guidelines. Like if we are using vaccines, we are using some viral part to another species, or this category includes research which have very high risk. Okay. So this is the biosafety 4th level, or it also includes the pathogens for which we don't have any kind of drug or vaccines till now. Fine. So this takes under category 3. So let's see why these guidelines are important and why DBT have projected these guidelines. First, these guidelines regulate research on GMOs and transgenic organisms, that how many researches on what GMOs are produced by these researches. So they keep an eye on it and they make a database for this. Second one is ensure biosafety and risk assessment in genetic engineering. Ensure biosafety, that what gene is coming to the community or the society in the form of food or plants, or in maybe animals, and what is the risk associated with these genetic transformations. So the third one is classify experiments based on risk level. So we have to check that our experiment or our research falls under which biosafety level, and we have to take their approval according to that before commencement of our experiment or before release of our GMO product. So here are some links which I have provided. You can go on these links and check these guidelines in more details. So let's see some of the updates after 1990. In recent years, India has updated its regulatory framework concerning recombinant DNA technology to enhance biosafety and align with international standards. So there are some notable amendments which I am going to show you. First is Biosafety Guidelines, which are amended in 2017. What are these? So in 2017, the Department of Biotechnology's Review Committee on Genetic Manipulation, in short RCGM, released comprehensive guidelines titled Regulation and Guidelines on Biosafety of Recombinant DNA Research and Biocontaminant. These guidelines addresses biosafety in rDNA research and the handling of hazardous microorganisms and genetically engineered organisms or cells. So they emphasize stringent facility structures for handling various organisms. The institute which is carrying out research should have proper facilities to carry out research to handle those microorganisms before starting of the experiment, and provide clear instructions on waste disposal, decontamination and emergency procedures. So the guideline also include a list of risk group agents and determine appropriate contaminant level for handling in India itself. So now here comes new drugs and clinical trials rule in 2019. So the Central Drug Standard Control Organization introduced new drugs and clinical trial rules, streamlining the approval process for new drugs and clinical trials. So this includes rDNA technology, and these rules aims to ensure faster approval while maintaining stringent safety and efficiency standards. Then in 2024, updated guidance for industry for biologicals is introduced. So in May 2024, after 16 years, the CDSCO, that is Central Drug Standard Control Organization, released an updated version of the guidance for industry for biologicals. This update aligns with current regulations and incorporates online application processes, reflecting advancements in biotechnology and regulatory practices. Okay. So in short, in 1990 guidelines, it is written that regulate research on GMO and transgenic organisms. They ensure biosafety and risk assessment in genetic engineering and classify experiments based on risk level. So if you find this video informative, then please like, share and subscribe, and press the bell icon for more notifications. And in another video, I am going to tell you the guidelines of 1998. So make sure that you are subscribed so you will get the notification for that. Stay tuned. Meet you soon.